I welcome you in this lecture. Well, I am Dr. B.P. Oli. I am working as Assistant Professor in the Department of Zoology, Swami Vivekanand Government PG College, Lohagat, District Champawat. The title of lecture is Mutation, its historical background, chromosomal mutation, chromosomal aberrations. Let us start the topic. Chromosomal mutations and chromosomal aberrations both refer to sudden and discontinuous changes in the structure and number of chromosomes. Or mutation may be defined as large or small change in the genetic mechanism of an organism. Mutation may occur both in somatic as well as germinal cells. Somatic mutations are acquired and insignificant from the evolutionary point of view, whereas those of germinal cells, means sperms and ova, are heritable and have evolutionary significance, known as germinal mutations, and form the raw material of evolution. Uh, Here is brief historical background of how the understanding of mutations developed. Early evolutionary ideas in 19th century. First of all, Charles Darwin in 1859 in On the Origin of Species, Darwin proposed the theory of natural selection, suggesting that species evolve over time through the differential survival and reproduction of individuals with favorable traits. Although Darwin did not use the term mutation, he recognized that variation among individuals in a population was crucial for evolution. Then Gregor John Mendel in 1865, Mendel, also called the father of genetics, he conducted experiments with pea plants and discovered the basic principles of heredity. His work on inheritance laid the foundation for understanding how traits are passed from one generation to the next. But he did not know the source of new variations or mutations that could give rise to new traits. Second is early mutation theory during late 19th to early 20th century. Hugo de Vries in 1901, he propound the concept of mutation as a source of genetic variation, and it was given by Dutch botanist Hugo de Vries while working with the evening primrose Oinothera lemarchiana. This is the scientific name of that plant. He observed sudden distinct changes in traits from one generation to the next and coined the term mutation to describe the changes. Hugo de Vries proposed that mutations, rather than gradual changes, were responsible for new species. Then Thomas Hunt Morgan in 1910. Morgan work with fruit flies, Drosophila melanogaster, demonstrated that mutations occur in genes and can be inherited. He linked mutations to changes in chromosomes and genes, showing that they could produce new traits that could then be subject to natural selection. The modern synthesis, starting from 1930 to 1940, the modern synthesis of evolutionary biology integrated Darwinian natural selection with Mendelian genetics. Scientists like Ronald Fisher, J.B.S. Haldane, and C. All Wright recognized mutations as the source of genetic variation, while natural selection acted on that variation to drive evolution. Mutations were understood as random changes in the genetic material that could be neutral, beneficial, or harmful. Then molecular genetics from mid 20th century onwards DNA structure in 1953, the discovery of double helix structure of DNA by James Watson and Francis Crick, along with Rosalind Franklin's work on X-ray diffraction, revolutionized the understanding of genetics. The structure of DNA explained how genetic information is stored and how mutations could arise as changes in the DNA sequence. Understanding mutations, with advancements in molecular biology, scientists began to understand mutations at the molecular level. Mutations were seen as changes in the nucleotide sequence of DNA, including point mutations, means substitution, insertion, or deletion of bases, chromosomal mutations, such as inversion or translocations, and larger genomic alterations. These changes could be caused by errors in DNA replication, environmental factors like radiations or chemicals, or spontaneous events. Next is modern understanding during late 20th century to present. Today, mutations are understood to be the fundamental source of genetic diversity and a key driver of evolution. They can be beneficial, neutral, or harmful depending on their effect on the organism. Molecular techniques like DNA sequencing have allowed scientists to study mutations in unprecedented detail, including their roles in cancer, genetic disorders, and evolution. The discovery of CRISPR gene editing technology has allowed scientists to induce specific mutations in genes, offering potential treatments for genetic diseases and further understanding of gene function. In nutshell, the concept of mutation has evolved from early ideas about variation and inheritance to a detailed molecular understanding of how genetic changes occur and affect organisms. Kind of mutations on the basis of origin of mutations, they can be categorized as follows. First is spontaneous mutation. These are naturally occurring mutations. Their origin is unknown. For example, polydactyly in man, sheep, cats, albinism in man, pig, mice, hornless cattle, hares, lip in man, lobster claws in man, double-eared cattle, and multi-nippled condition in sheep. Second is induced mutation. Any mutation which is produced in any living organisms in the laboratory by mutagenic agents. Kind of mutations on the basis of the nature of their effects. First, morphological mutations. Mutations affect visible properties of an organism like shape, color, size. Example, curly wings in Drosophila. Second is lethal mutations. Here, the new allele is recognized through its lethal effects on the organisms. Next is conditional mutation. In this class, a mutant allele expresses the mutant phenotype under a certain condition called the restrictive condition, but expresses a normal phenotype under another condition called the permissible condition. For example, in permissive condition, the Drosophila are normal at 20 degrees centigrade, but die off if the temperature is raised to 30 degrees centigrade, means restrictive conditions. Then biochemical mutations. This class is identified by the loss of or a change in some biochemical function of the cell. Next is resistant mutation. Here, the mutant cell or organism acquires the ability to grow in the presence of some specific inhibitor, such as pathogen or cyclohexamide, to which wild types are susceptible. Last kind is spurious mutation. Uh, there are other type of mutations which remain suppressed but manifest themselves in the offsprings as a result of crossing over. For example, pink eye coloration in Drosophila. Uh, According to the different directions in which mutations can appear, we have the following types. Forward mutation. Those mutations which result in the development of abnormal characters from the wild characters, normal, are called forward mutations. Such mutations always produce deviation from normal main stock. These are more common in nature. For example, vestigial wings in Drosophila. Second is reverse mutations. Sometimes the mutant phenotype are suddenly changed again to wild type of phenotypes. Such reversion from the mutant to the wild type is called reverse mutation. For example, in a population of vestigial winged Drosophila, suddenly a long winged fly makes its appearance. Next is the kind of mutations according to the types of chromosomes. First is autosomal mutations. Those mutations occur in autosomes. For example, Down syndrome in human being. Second is sex-linked mutation. These are such mutations which occur in sex chromosomes, X or Y chromosomes. If mutation occurs in X chromosome, then it is called X-linked, and if in Y, then Y-linked mutations. For example, sex-linked diseases. Kind of mutations according to the stage at which they occur. First is gametic mutations. When mutations occur during the gametic formation, resulting in the development of the mutant character in all the gametes formed. Another is zygotic mutation. When mutations occur during the first or later mitotic divisions in zygote, this results in the development of mutant character in only those cells which are involved during the process. In these cases, a mosaic organism is formed. Now the next category is kind of mutations according to the affecting factors. There are two types of mutations according to their influencing factors. First is endogenous mutations. These are caused by certain internal factors like change in metabolism, nutrition, certain radioactions of internal origin, and so on. Lack of proper nutrition has been found to cause mutation in bacteria, fungi, etc. Second kind is exogenous mutation. These mutations, which are caused by external factors, they are caused by change in temperature or climate or environment, etc. Nature of mutations. Mutations can occur only when there is any sudden change in the nature of gene or in the structure of chromosome or in the number of chromosomes. Thus, broadly speaking, mutations can be first categories chromosomal mutation or mutation due to chromosomal aberration. Second category is gene mutations, and the third is genomatic mutation. Now, chromosomal mutations. Chromosomes are delicate structures of a cell that can break due to ionizing radiation, physical stress, or chemical compounds. Chromosomal mutations refers specifically to changes in the structure or arrangements of chromosomes in an organism. These changes can affect the location or number of genes on a chromosome and usually result from errors during cell division, particularly during meiosis. Chromosomal mutations typically involve large sections of DNA and can have significant impacts on an organism's traits and developments. Uh, Types of chromosomal mutations. First is deletion. When a part or segment of the chromosome containing some genes is lost or deleted, leading to the loss of several genes. For example, in this figure, the broken ends of the chromosomes have been described as sticky ends and can be reunited by the enzymatic process of the cell. In first end of the chromosome, A part is lost, while in another end, G is lost. Hence, they form a ring chromosomes. Next category is duplication, which involves addition of a part of chromosomes. Third category is inversion, when a segment of the chromosome is reversed end to end, altering the gene order. And fourth is translocation, which involves exchange of segments between non-homologous chromosomes. These are two types of translocation. First is reciprocal, where two chromosomes exchange segments, and second category is non-reciprocal, where only one chromosome donates a segment. The chromosomal aberration is broader and refers to any structural or numerical abnormality in chromosomes, encompassing chromosomal mutations as well as changes in chromosome number. Chromosomal aberrations can be structural, altering the shape or makeup of the chromosome, or numerical, altering the number of chromosomes in a cell, often caused by errors in cell division, means non-disjunction. Common forms include monosomy, when a chromosome is missing from a pair. For example, Turner syndrome, where there is only one X chromosome. Second category is trisomy, when there is an extra chromosome. For example, Down syndrome, where individuals have 3 copies of the chromosome 21. Third category is polyploidy. There are more than 2 complete sets of chromosomes, common in plants, but usually lethal in animals. Causes of chromosomes are delicate structures of a cell that can break due to ionizing, radiation, physical stress, or chemical compounds. Besides these, sometimes errors during cell division, for example, non-disjunction during meiosis, causes mutations. These are the some factors which causes the chromosomal mutations or aberrations. There are few examples or conditions caused by chromosomal mutations. First is Down syndrome, trisomy 21, a numerical chromosomal aberrations, where there is an extra copy of chromosome 21. Another example is Turner syndrome, monosomy X, a numerical aberration where there is only one X chromosome. Next is Cry-Du-Chet syndrome, structural chromosomal mutation caused by a deletion on chromosome 5. Next example is chronic myelogenous leukemia, commonly called CML, often caused by a translocation between chromosome 9 and 22, which is also known as the Philadelphia chromosome. Here are few substances which are found in our food materials causing mutations. A number of substances are taken with food knowingly or unknowingly which causes mutations. Some of them are discussed below. First is nitrosamine. It contains sodium nitrate, has been very widely used both as a preservative and a color enhancer in meat and fish products. It is highly mutagenic. Another is artificial sweeteners. It means sodium cyclamate has been widely used as a sugar substitute in diet, foods, and soft drinks. But sodium cyclamate has been a carcinogenic effects in the bladder of mice. Artificial sweeteners like saccharin are used to replace cyclametone. Next is caffeine. Caffeine produces mutations in bacteria as well as in fungi. Coffee itself can produce carcinogenic effects. Other food materials are aflatoxins, PAHs, means polycyclic aromatic hydrocarbons, excessive use of alcohol, pesticide residues, mustard gas, colchicine, etc. The rate of mutation may be defined as in several different ways. It usually expressed in terms of mutations per cell individual or generation. For example, among fruit flies, about 4 sperm cells in every 10 is power 5 will carry a mutation for red eye to white eye. Now, the importance of mutations in industry. By treatment with mutagenic agents in combination with genetic methods, it has been possible to produce the Penicillium chrysogenum strain, which is 1,500 times more productive than the original strain. Besides, many useful varieties of edible plants, fruits, domestic animals, yeast, etc., have been obtained by induced mutations. Now, here are few references which I have consulted. A book, Aurora and Sandhu Genetics, published by Himalaya Publishing House, India in 2012. Then Gardner, Simmons, and Snutstead, 2014, Principles of Genetics, Ville India Private Limited. I have also consulted many websites and net materials during this preparation of this lecture. Thank you.